We've talked a lot about ribonucleic acid, or RNA. Now we're going to talk about specifically non-coding RNA. We know a whole lot about messenger RNA. Messenger RNA carries the gene's message from the DNA to the ribosome. But there's a whole lot of RNA that does more than just that. Protein-coding DNA sequences account for only a small fraction of the human genome, less than 2%. So if you're looking at this graph, it probably should have a legend with it, because it really looks like it's saying that there's a tiny fraction of the genome that's not used for coding. It's actually showing that blue section there is the amount of the genome not used for coding. The rest of the sequences are used for other things. The RNA produced by all of that, the rest of that DNA, has distinctly different functions. There's three big types of RNA we've talked about: ribosomal RNA, transfer RNA, and messenger RNA. But there's some other non-coding RNA that's used in transcriptional regulation: microRNA and small interfering RNA, or otherwise known as siRNA. MicroRNA is complementary to some coding strand of RNA, causing them to bind. So if you have a piece of RNA, it's got a complement with this microRNA. The two will touch each other and stick. This creates double-stranded RNA. Well, the cell recognizes double-stranded RNA as a virus or viral RNA, so it immediately gets to work cutting it apart. If the RNA is cut apart, it can't go on to produce a protein, so transcription is decreased due to the lack of messenger RNA. This process is called RNA interference, or RNAi, and it's used to turn off genes. Small interfering RNA functions very similar to microRNA. However, this small interfering RNA can also bind to DNA, and that turns the DNA off. So the RNA binds but never lets go. That turns the genes off that would otherwise have been transcribed. Non-coding RNA, the RNA that is not used for encoding proteins, is going to have an impact on not just other cellular processes, but specifically on genetic regulation. So even though they don't code for a protein, they are going to be responsible for making sure the protein gets expressed in the right quantity. So we've got lots and lots of different kinds. We've got microRNAs, we've got siRNA, snRNA. The non-coding RNA may bind to other RNA, causing it to degrade. It may serve as a way to help promote, serve as basically like a promoter to help recruit polymerase. There's all sorts of things it can do. So non-coding RNA that does not code for anything, can act on other RNA by binding to it, leading to that RNA being broken down so that the cell can regulate how much transcript, how much RNA is present, which means it'll be able to regulate how many proteins are being produced. The content review questions here are to focus your studies. In the next group of lectures, we're going to be talking about biotechnology, so applications of the central dogma of molecular biology and regulation, as well as delving into the ancient past and what is historically been known as biotech. So yeah, it's kind of bleeding edge technology. We'll talk about it in a second.